New anatomical findings in the foveola may explain the Stiles-Crawford effect number 1. A human macula consists from a fovea with a diameter of 1.8 millimeters, and in the center of the fovea there is the so-called foveola, which is only 350 microns in diameter. Also, the macula exists since 550 million years. The anatomy of the foveola is more or less still unknown, so unbelievable it is. What we did, we investigated about 50 foveolas by electron and light microscopy. We performed serial sections from 8 foveolae of humans and monkeys, and we mounted the image stacks and segmented them, and this resulted in 3-dimensional models. Here we see a cross section through the foveola of a monkey eye, and in the center of the foveola we find very unique prominent Müller cells. They can be recognized as white dots. Here the diameter of this area is around 100 microns. In the next slide we see the same cells at high resolution in an electron micrograph. Here we have only 2 types of cells. We have the photoreceptor cells, here the nuclei of the photoreceptors, the handle fibers, and in between these very large Müller cells. Interestingly, these Müller cells do not contain any cell organelles. This is very, very unusual, but we think this is necessary for light guidance through these cells. Physiologically, these central Müller cells may be related to the Stiles-Crawford effect, which was regarded as one of the most important discoveries of the last century and was discovered 84 years ago, but still is not understood. The Stiles-Crawford effect describes that a light beam entering the eye directly is much more sensitively perceived than the same light beam entering at an angle as a function of the pupil location. And here you can see the relative luminance measurement in a human eye, and you can see that we have a sensitivity drop dramatically from the central part of the pupil to the margin of the pupils on both sides. We constructed 3-dimensional models from human retinas because we wanted to see whether they also contain large Müller cells, and here you can see some of them already in this frontal view. Such an image stack consists from more than 150 individual sections, and here you can see an individual Müller cell in white that after isolation shows a plateau zone at the top of the retina. And we think that this plateau zone may play a role in causing the Stiles-Crawford effect. Light that hits the Müller cell plateau directly is transmitted into the photoreceptors. In contrast, light that hits the Müller cell plateau under an angle is partly reflected, which reduces the sensitivity of the photoreceptor cells. To test this hypothesis, we used human foveae on cover slides and changed the angles of the transmitted light from 0 to 10 degrees. This is what you can see. We have now we have around 5 degrees, now we have 10 degrees. The central part becomes completely dark. We go back. Now we may have probably 5 degree and now 10 degree, and it is completely dark. This what you see here is the yellow spot is the macula pigment. And we also quantified the intensity of the light reflection at 0, 5 or 10 degree, and we found an intensity curve which was very similar to that what was measured by Stiles-Crawford in 1933. In conclusion, in the foveola, unique Müller cells are present, which probably caused the Stiles-Crawford effect, number 1, by angle-dependent reflection of the incident light. Thank you very much for attention.